Have you ever heard someone say we've got a brain microbiome? Not just the microbiome influencing the brain from a distance, but actual microbes living inside the brain tissue itself. Stay tuned to find out more. Every so often, studies pop up claiming they found microbial DNA or even microbe-like structures in the human brain. A few researchers have wondered if the brain might host its own hidden microbial community, something completely outside the traditional view of neuroscience. Others insist this is impossible. So, in this video, we're going to break it down. 1. Does a true brain microbiome actually exist? 2. If not, why do some studies keep finding microbial traces in the brain tissue? And 3. If the brain is microbe-free, how do microbes still end up shaping our thoughts, emotions, and behaviour? Let's dig into what the evidence says. Okay, so here's a quick refresher. Microbiomes and the gut-brain axis. A microbiome is the collection of microbes, so bacteria, fungi, viruses and so on, plus their genes and products, living in a particular environment. Your gut, your skin, your mouth, your lungs, and various other places. Now the gut microbiome is the celebrity here, so we now have evidence that gut microbes can talk to the brain via the vagus nerve, a long cranial nerve linking the gastrointestinal system to the central nervous system. They produce metabolites like short-chain fatty acids and neurotransmitter precursors. They modulate inflammation, which in turn affects brain function and mental health. Now, these mechanisms are supported by some evidence in humans and plenty of evidence in non-human animals. But that's very different from saying the brain itself is colonised by its own resident microbial community. So for decades, the dogma has been that the healthy brain is sterile, thanks largely to what's called the blood-brain barrier, essentially a very picky bouncer deciding what gets access to your neurons. So why did scientists think that the brain was sterile? Well, the sterile brain idea became so entrenched because the brain is incredibly vulnerable to infection. Meningitis or encephalitis are obvious and devastating when microbes invade. Now, the blood-brain barrier and immune surveillance are extremely strict. Microbes seen in the brain are almost always associated with disease. So, historically, the brain was treated as an organ where microbes must not be allowed. Then, high-sensitivity DNA sequencing arrived, and everything got messy. The controversial claims: we found a brain microbiome. So over the past decade, some studies have reported microbial DNA or bacteria-like structures in the brain tissue. For example, a 2016 study of multiple sclerosis lesions reported bacterial DNA and bacterial cell wall components within multiple sclerosis plaques, raising questions about whether microbes or microbial fragments can access the brain in disease states. A widely discussed 2018 conference abstract from the University of Alabama at Birmingham presented electron microscopy images of bacteria-like structures in post-mortem human brains, sparking speculation about a possible brain microbiome, although these findings remain preliminary and have not been confirmed in peer-review follow-up work. This research suggested there might be resident brain commensal microbes. These microbes could influence neuroinflammation or neurodegeneration. It's a seductive idea, but there's a catch. When a sample contains almost no microbial DNA, known as low biomass samples, like brain tissue, it becomes incredibly easy to fool ourselves. Even the lab kits, tubes, and chemicals we use for DNA sequencing have tiny traces of microbes on them. If those get mixed up into samples, the computer can mistake the background noise for a real microbiome. In other words, when scientists are working with tissues that should have very few microbes, it's surprisingly easy to create a fake microbiome by accident. This exact issue plagued research on blood, amniotic fluid, and the placenta. And when brain data are reanalyzed with strict contamination controls, the brain microbiome signals often disappear. Most experts now conclude there is currently no convincing evidence for a stable, healthy brain microbiome or community of microbes in the brain. The contamination problem. Lessons from the placenta. A really good example comes from the placenta. A few years ago, some big studies claimed the human placenta had its own microbiome, meaning babies might be exposed to microbes before they were even born. It made headlines at the time, but when other scientists repeated the work using much stricter controls, they discovered something important. almost all the microbes were actually coming from the lab kits, the chemicals used in the testing, or from the birth process itself. Once those sources of contamination were removed, the supposed placenta microbiome essentially disappeared. In healthy, full-term pregnancies, there's currently no solid evidence that the placenta has its own resident community of microbes. Experts now warn, low biomass samples demand ultra-strict controls. And when you apply these to brain tissue, the evidence for a brain microbiome also evaporates. So, today, the consensus is the healthy human brain is very likely to be microbe-free. Not because microbes never get in, but because the brain doesn't host a structured resident community. But microbes do get into the brain, particularly in disease states. It's important to separate two things. One, infection. Microbes invade the brain and cause pathology. And two, microbiome, a consistent, dynamic and often beneficial community that actively lives there. We've known for a long time that microbes can enter the brain in disease. Things like bacterial meningitis, viral encephalitis, herpes simplex virus linked to dementia risk, fungal meningitis, and periodontal pathogens detected in Alzheimer's brains. But those are essentially invasions rather than a microbiome. The oral microbiome-brain connection. Here's where a real and very interesting story emerges. Even if the brain doesn't host its own microbiome, we think the oral microbiome can affect brain health, without physically colonising the brain. Scientists now know there are several clear ways the oral microbiome, i.e. the community of microbes in your mouth, can influence the brain, even though the brain itself doesn't seem to host its own microbiome. Here are the 3 best supported pathways. Number 1. Inflammation and immune signalling. So gum disease, periodontitis, doesn't just affect your mouth. It creates a kind of slow, chronic inflammation throughout the whole body. Research shows that certain oral bacteria, like Porphyromonas gingivalis and Fusobacterium nucleatum, can raise inflammatory molecules in the bloodstream, such as IL-6 or interleukin-6 and TNF-alpha. These inflammatory signals can affect the blood-brain barrier, influence the brain's immune cells, also called microglia, and increase overall neuroinflammatory stress. And long-term inflammation is strongly linked to depression, cognitive decline, and faster brain aging. 2. Microbial products reaching the brain, not whole microbes. You don't need bacteria to physically enter the brain for them to have an impact. Oral pathogens release toxic molecules such as LPS, or lipopolysaccharides, a strong inflammatory trigger, and gingipains, enzymes made by P. gingivalis. A study by Dogma and colleagues in 2019 found gingipains in the brain of people with Alzheimer's disease. In mouse experiments, these molecules could trigger amyloid beta production, one of the hallmarks of Alzheimer's. This suggests a pathway like gum infection to inflammatory molecules in the blood to effects on the brain tissue, without any actual bacteria needing to colonize the brain. Vascular and metabolic pathways. So periodontal disease is also linked to problems with blood vessels and blood flow. Reviews have shown that gum disease increases the risk of stroke, atherosclerosis, so narrowing of the arteries, and vascular dementia. Poor brain blood flow is one of the strongest predictors of cognitive decline. Now, there are also some suggestions of oral microbes passing the blood-brain barrier, but this is still considered rare, disease-linked, and not evidence of a normal brain microbiome. Most studies that have detected oral bacteria in the brain have done so in the context of infection, chronic inflammation, or neurodegenerative disease, not in healthy people, and the mechanisms are still being investigated. So what about mental health? So there's now fairly good evidence that problems in the mouth, especially gum disease, are linked to conditions like depression and anxiety. Reviews have found that people with periodontitis were more likely to experience depressive or anxiety-related symptoms. So why might this happen? Well, researchers think a few things could be going on. Number 1, inflammation. So gum disease creates ongoing low-grade inflammation in the body, and inflammation is strongly linked to changes in mood and brain function. 2. Pain and stress. Living with chronic mouth pain affects eating, sleep, and social confidence, and this can wear people down emotionally. 3. Changes in diet. If your mouth hurts, you may avoid crunchy, fibrous foods, which can indirectly affect gut health and overall well-being. 4. Microbial byproducts. Molecules released by oral bacteria can travel through the bloodstream and influence the immune system, which is tightly connected to mental health. Importantly, this doesn't mean oral bacteria are living in the brain. It means that what happens in the mouth can influence the rest of the body, including the brain, through inflammation, immune signalling, and other whole body pathways. The mouth and brain are connected, even though microbes don't necessarily travel up into the brain and set up a colony there. So what should we do about it? Well, evidence-based steps to support oral brain health include good oral hygiene, treating periodontitis early, a diverse diet rich in polyphenols, avoiding smoking, possibly trying probiotics that may reduce oral inflammation. Now, these are practical data-supported strategies to reduce systemic inflammation and protect brain health. Now, there's another interesting story: remote control from the gut. So you don't need microbes living in the brain for them to shape how you think and feel. Your gut microbiome already communicates with the brain in several powerful ways. So gut microbes can affect brain chemicals. They help produce building blocks of serotonin, dopamine, and other neurotransmitters. The immune system, which strongly influences mood, stress, and inflammation in the brain. Stress and anxiety responses, by interacting with hormones and the vagus nerve. Microglia, the brain's own immune cells, which guide brain development and repair. The blood-brain barrier. They can make it slightly tighter or leakier depending on the state of your gut microbiome. Now, microbes also make important metabolites like short-chain fatty acids that can travel through the bloodstream and influence how brain cells work. So, even if the brain itself stays basically microbe-free, it's still deeply connected to the microbial ecosystems living elsewhere in your body. Understanding how and when microbes breach the blood-brain barrier could open new insights into Alzheimer's and multiple sclerosis, etc. But the strongest evidence today supports this. The brain is unlikely to host a microbiome, but is profoundly influenced by microbiomes elsewhere, especially in the gut and the mouth. Stay open-minded though, but demand rigorous evidence. If you found this episode helpful or interesting, please do me a huge favour and like and subscribe to the channel for more science-based insights into how biodiversity, including the invisible kind, shapes your health. And please sign up for my free newsletter over at www.jakemrobinson.com. See you next time, and remember, stay wild inside and out.